Good morning, everyone. I'll keep this fairly short and sweet. My name is Andrew Hessel, and as George just pointed out, I've been thinking about whether it's time for a new human genome project for some time. I wrote that little piece in Huffington in 2012, but it wasn't the first time I had thought about it. My history in genomics started really quite early on, and not as early as George. Most people, a lot of the early history of the first human genome project has kind of been forgotten, but it was a really radical idea that started to be proposed back in 1984. It started to pick up some energy in 1985 as people came together and started talking about the technical feasibility of the project, and then starting to consider whether it should be done, and of course, how the project would be funded. I first heard about it really when it launched, when it started to get the attention in 1990 with James Watson and the various folks and the large dollars being thrown into genomics. And really, it inspired me to work in the area of genomics. I don't know what your entry point into this field has been, but really it was the idea of being able to sequence organisms and look at their code and really the instruction set for these creatures that just completely thrilled me. And at the time, it was a slow process to read DNA. It was difficult to analyze and interpret. But it was just incredible watching the acceleration of this technology over the next decade, and it really put this science into the public perception. Of course, there were debates about the technology and how it could be used. It turned into an incredible race between public and private, and some very charismatic and newsworthy scientists. But it made it accessible to the public. And of course, when President Clinton brought the public and private groups together and essentially declared the draft genome complete, I think it was at 93% or something at that point, but still is. That was kind of a moment of closure, at least for the public. There was still, the work continued, is continuing, but now sequencing is just taken for granted. So what was incredibly provocative, pushed everyone's capabilities, and made careers, inspired, humanity, really fell off into a quiescent period. Over the last, since that time in 2000, we've seen the emergence of a new technology, being able to write DNA faster and cheaper. And this is, it's shaped my career as much as the first human genome project. At the time, I was working for a biotech company, and they said, well, reading the human genome is essentially complete. Why don't we start thinking more about writing technologies? And they weren't interested. It wasn't a bottleneck for their drug development processes. So I thought, well, I don't want to work for a biotech company that doesn't want to write DNA. So I left the field and just started to explore the world of synthetic biology. And I've had the great pleasure to watch this field emerge over the last 10 years in particular. And I know many of you in the room and the work that you do. I don't know where it goes. All I know is we're at very early days of synthetic biology. And today I do a lot of public speaking. Most people haven't heard about writing DNA. They have very little understanding about what the potentials of this technology are, what it means to society, what it means to medicine, what it means to humanity. This has always disappointed me. It means either as scientists we're not doing a very good job communicating. It just may mean, might be we just need a better marketing department. In 2009, I joined Singularity University. I wrote the life science program, the initial draft of it. And this is a group of people that are really forward-thinking, that propose dangerous ideas every 20 minutes. And in my first presentation to the executive program that I was teaching, I mentioned it's probably time to write a human genome. And they said, you probably shouldn't talk about that for the moment. It's a little too dangerous an idea. So I put it on the back burner. When I wrote the article for Huffington Post, there was just silence. And up until a few weeks ago, if you Googled human genome synthesis, you basically got nothing. There's a void out there. Yet it's that we will go down the path of writing more and more complex genomes, and ultimately human seems to me inevitable. It's a journey that we're exploring here today. It's not going to end here today. This is the start of many different things, I'm sure. But I just want to say thank you for having an open mind. Thank you for being here. Thank you for bringing your skills to the table. And I hope in 10 or 15 years, when we look back at this moment, we realize that we might be making history today.
Welcome. I'm more of a detail guy. So if you could cue the slides, please. Oh, there we go. I'm going to offer up some thoughts on HGP-write, as we like to call it, designing and synthesizing modified versions of human genomes. And I need to disclose my engagement in a couple of companies here before I talk. So I think one of the biggest questions on everyone's mind, and it certainly was on my mind the first time Andrew sprang it on us last July, was why and should we synthesize a human genome? And I add to that in cells, and that's a very important addendum. So just a few thoughts before we dive into the details about what we've been discussing and we hope you will discuss with us. As you've heard already, we seek to do this responsibly and not using sort of so-called deficit model thinking. Deficit model thinking, I've learned, is telling people why what you're doing is important and then going off and doing it. So a good example of this is the widespread rejection of vaccines by a substantial proportion of our population. They were told it was important, but there was no real engagement or outreach at all. And in contrast, a good example of responsible innovation involves the so-called arsenic biosensor that was developed as one of the first synthetic biology projects, where the people involved in the project widely engaged government officials, NGOs, and most crucially, the potential end users really understood the problem, heard them. Another really important point, as I said, we are talking about doing this in cells, lest there be any confusion on this, i.e. somatic applications. So we don't anticipate that there's going to be a major discussion about germline modification here. That discussion is already out there, and we have decided that our involvement in this is strictly somatic. So I'm a skeptic by nature, and it took me a while to warm up to this project. But over the last few months, I've recognized several big drivers for me to say yes and not only enthusiastically participate, but really be a proponent of this. I mean, obviously, this is an incredible tool for learning fundamentals of genomes. As you've already heard, technology development will come out of this and have many positive spin-offs beyond human genome for applications to global problems. For example, gene drives could revolutionize health in the tropics, Zika, malaria, dengue, chikungunya. Crop engineering could improve food security through pest resistance, etc. And the field of synthetic biology obviously is to some extent limited by how rapidly and efficiently we can make DNA and put it to use. And finally, I'm at a medical center, so the biomedical somatic applications seem very real to me. For example, one can imagine engineering universal T cells or T cell therapies, revolutions in organ and tissue transplantation, we'll hear a bit more about later today, and perhaps a next generation of gene therapy. Some of my colleagues and I have been talking about a stepping stone project. I'm a basic scientist at heart, and one of the most fascinating aspects of the human genome today is that we've discovered all of these loci on the map that are associated with disease and other human traits, but we really don't understand them because these variations in our genome lie in the so-called dark matter in between the genes. So here's a picture of a human gene. And I don't know if there's a pointer here. As most of you know, human genes are made up of tiny little blocks. They're split by much larger regions called introns. And then if we zoom out, here's that same gene, we see very large gaps between the genes. And thanks to large support by the NIH, there's lots of clues about places in the dark matter that are important for controlling the expression of these genes, and therefore a better understanding of how they work and how the cause of disease. So we're actually proposing a project we call a stepping stone project right now. Here's a gene, let's call it your favorite gene, where we would make 10, 100 to 1,000 synthetic variants of that gene, assembling these in a yeast cell, and then incorporating that very efficiently into a specific human cell line so that we would be able to then evaluate the specific effects of each of these little variations in the dark matter on the expression of the gene and ultimately the function of that gene. So how should we start a larger project to understand the human genome? You'll hear about pilot projects, and we've decided to sort of promote the idea of 1% of the human genome, but a very informative 1%, not a random 1%, as has often been done in other genome projects, including our Synthetic Yeast Genome Project you'll hear more about. These are designed to teach us new biology, develop important tools for therapy, explore new directions, and engineer other non-human genomes. And in parallel with this, technology development will proceed and make the ultimate synthesis of a human genome much more practical. So new technology, new tools, new ways to handle big DNA, integrate it, better defined cell lines, et cetera. And very importantly, in order to do this efficiently, we will need to really employ automation in order to make this really fly. And so this is our Genome Foundry that we are setting up at NYU showing how we're automating one bottleneck in this process. So where do we start with this? As I've said, we're talking about a cell line engineered for safety. This would be presumably an iPS cell so that it would have the capability of being differentiable into different types of tissues. And it would be engineered to have a strict germline firewall. For example, removal of one of the sex chromosomes. So an EXO cell line would be a simple way to assure that this would not be able to go germline. And these cells can be differentiated into tissues and organoids, models of human organs, to evaluate the functional effects of any designer changes that were introduced. And here in Boston at the Wyss Institute, for example, there are these so-called organs on chips that provide excellent models for lots of processes that happen in the body. So how would we do this? Well, this is obviously a longer discussion, but depending on what exactly we plan to do, that will dictate the ultimate design. 1% pilot, you'll hear me talk about one of those in a bit, might engineer just these short blocks or exons, which are very small. These will require relatively small pieces of DNA, but not giant pieces of DNA. Whereas if we were to really do an entire genome synthesis, it might benefit from a strategy that you'll probably hear about a little bit later on, used to synthesize a modified version of the yeast genome called Swapin, where potentially 100,000 DNA letters at a time could be introduced and used to replace the endogenous 100,000 DNA letters. So to make a 3 billion base pair genome would require 3,000 rounds of this. Now that sounds like a lot, but it's not inconceivable by any means. And surely there are many ways to make this more efficient. One very interesting publication that just came out recently was the development of haploid stem cell lines, which could make projects like this much easier. And we'll hear a little bit about those later today. So to wrap up, there's two last points I'd like to make, and they have to do with community. And the first one is, we don't think this should be a human genome project to the exclusion of other organisms. As the leader of the Yeast Genome Project, I'm keenly aware of this and a huge proponent of appreciating our biological diversity and bringing some of this technology to the many other organisms on the planet for practical reasons and for reasons of dissecting the fundamental biology of the system. For example, I think we're already learning a lot from microbial genome engineering. And secondly, the scientific community, this is a grand challenge, and as such, it really demands global participation. We have interested parties here today from Australia, China, Singapore, the UK, and more. Personally think this is a fantastic way to educate an international workforce in genomics to help promote the understanding of the field. And in the words of Huanming Yang, the chair of BGI, or Beijing Genomics Institute, as it was formerly known, it's more fun to do it together. We make new friends. Thank you. I'd be happy to take questions, and I'm sure my colleagues would as well. Stunned silence.
I think that the EXO
Right. It's one. There are versions of Turner syndrome where the whole X chromosome is not deleted, but as far as I know, when it's an XO, it's infertile. Yes, Richard. We can't hear. Sorry.
I just wanted to get your and George's opinion on how easy it would be to hack that firewall.
To hack that firewall? Well, that's an important question. Of course, the details of how easy it would be would depend on the details of exactly how that firewall was constructed. I can't really answer that, but obviously, since biosafety is an integral part of this, we're going to hear about that later on today. I expect that the firewall will be improved as the project progresses and will get better and better. But yeah, the details of that are to be determined, and therefore the impregnability of it is a little hard to predict at the moment.
If you're going to hack, you can just do it from scratch. You know?
Yeah. Exactly.
So good morning. Thanks for the chance to talk. So I'm a postdoc in Jeff's lab, and I've spent the last 5 years thinking about all aspects of this SC2 project, the Synthetic Yeast Genome Project, as has my colleague Patrick Tsai, a former postdoc with me and now at Edinburgh. So today we're just going to give you some highlights of this project and the lessons we've learned that I think are really quite applicable moving into larger and larger genome scale synthesis projects. So just to get going, the goal of the SC 2.0 project is quite simply stated as to build an entirely synthetic designer genome to power growth of our favorite single-celled organism, Saccharomyces cerevisiae. So not just a rewrite, this is actually a redesign and synthesis of something new. And so at the outset of this project, there were three design principles with competing agendas that were set out. On one side of the coin, the idea is to build something that has a wild-type fitness, a healthy cell. But on the flip side, the genetic code powering it is going to be different and provide new properties, specifically increased genome stability and increased genetic flexibility. So how to do that? On the one side, the idea up front was to preserve genome content, or gene content, so no genes are deleted at the outset. Keep the non-coding DNA intact under the assumption we don't know enough about it. And then take a modular assembly strategy. So the yeast genome is composed of 16 linear chromosomes and 12 megabases of DNA. So to build these chromosomes piece by piece in individual strains, progressively building more and more DNA by chromosome into 16 strains, and then combine them together and monitor fitness along the way. And on the other side, probably the more exciting side, what are we changing? Deleting all repeats from the genome. This includes TY elements as well as subtelomeric repeats. Deleting all of the tRNA genes from their native positions and recoding them onto a neochromosome. And Patrick will talk more about that. He's led that project. Building in a watermarking system so we can understand the difference between synthetic and wild-type DNA, and recoding a stop codon to improve the genetic flexibility, and then a scramble-inducible evolution system that allows us to change, once the chromosomes are built, this order and orientation and content of genes. So this required a lot of thought in the design, and I'll just highlight a few points here. The design has been applied universally to all 16 chromosomes. All of those changes are built across all 16 chromosomes in a centralized editing process that typically by chromosome involved a yeast genetics expert in collaboration with a computational expert. And all of this was done in the context of custom genome design software built by Joel Bader, who's here today, called BioStudio, and this is just a screenshot of that. So the design is actually quite extensive, and this slide is a video of the editing of synthetic chromosome 5. And so each sort of frame of this video represents a single edit. And you can see as the genome progressively shortens in size, elements are being deleted. A variety of other elements have been sort of popped in along the way as we make base substitutions, deletions, and insertions, going from a 550 to a 520 kb chromosome with extensive modifications. So, in fact, the genome is now designed. It encodes, or it's missing 750 kb that was previously there. 215 kb has been recoded, and we've added 130 kb, yielding a genome, once built, that will be 1.1 megabases changed, and it's actually reduced in size substantially. So I'll highlight a few of the technical challenges that I think really are going to be important moving forward. One, we had to come up with a DNA assembly plan, and we did that by defining a hierarchy, starting with the design chromosome and splitting it into progressively smaller pieces, going from what we call megachunks all the way down to oligos. And these provide different entry points for having DNA synthesized, where early chromosomes were built from the bottom up, and with the decreasing cost of synthesis, we're now jumping in at the mini chunk and chunk level and having synthesis providers build those pieces for us. We've come on, Jeff mentioned this program called Swapin that allows us to progressively integrate more and more DNA by switching oxytrophies as we move along the chromosome. In the interest of time, I won't get into the details, but I'm happy to talk about that. We've had to figure out how to debug chromosomes, meaning when designer changes confer a fitness defect in order to comply with our design principle number 1, how to figure out what those changes were, which specific changes, and then revert them to wild type or make additional changes. And this is an example. The chromosome that I built, SYN6, had a defect of petites, if you're familiar with yeast genetics. And we tracked that back to a single recoding event within an essential gene that led to reduced protein production, presumably by a function, as we've learned, of RNA folding, as it turns out. And we've also had to figure out how to consolidate chromosomes. Given that we're building 16 individual strains, we need to put them together. And so we've come up with a strategy that relies on yeast mating, which is obviously a great strategy for yeast and not necessarily applicable to other organisms. And I can talk about that in detail later if you'd like. Another important component of this project has been the education component. And that really was spearheaded at Johns Hopkins, Jeff's former university, in the Build a Genome course, which engages undergraduates in this project, building fragments of synthetic DNA, anywhere from 750 base pairs now up to 10 kb fragments, that then feed directly into the chromosome construction effort. So these kids get an experience like grad school in the sense that they're having lab meetings, but also like biotech in the sense that they are, meeting their milestones. That is expanded to a franchise operation at Tianjin University, where the substantial portion of the DNA for synthetic chromosome 5 and 10 was built. And there's representatives from Tianjin here today. So what's the status update on this chromosome, or on this project, excuse me. So I'm showing you here the 16 linear chromosomes, as well as the new tRNA neochromosome Patrick will talk about, where the blue represents synthetic DNA integrated, and wild type is in yellow. While nearly all of the DNA has now been synthesized for the project, about 60% of it in total has been integrated, representing about 7 megabases of the 11.3 of the genome.
My name is Patrick, and I did my postdoc with Jeff a few years ago and moved to Edinburgh to start my own group in 2013. So I start with Jeff. I started a consortium internationally, and I coordinate the consortium. So just want to tell you a little bit. So today we have, about 10 universities over 4 continents working on this project, and this is probably one of the biggest synthetic biology project in public domain today. This is the map. So we mentioned there are different size synthesizing chromosomes, but there are also different universities like Pasteur, EMBL, which contribute to the project. So as you can see, this is quite a worldwide effort. So how we started? So a few years ago, I met YJ Yuan in Boston during iGEM. I think that was like 2011. And I know YJ for many years, and I talked to YJ, this is probably the next best thing going to happen in China, because YJ and I started iGEM in China in 2006. And then YJ took the project on board, and then we organized the first synthetic yeast meeting in Beijing with Huaming Yang. So this, is the picture we taken that day in 2012. We all look so young, but what we really do is we take many people who we think are going to be the key players in this project, like Tom Alice and people from France and Belgium and Hong Kong and India, And we also fly all the funding agencies, including BBSRC and Indian and funding agencies. We bring everyone into one room, kind of like what we are doing today, and have a discussion what should be done and how we're going to do it. And that was funded by NSF Savvy Science Across Virtual Institute, and that really stimulates the whole thing. So with each side, we sign agreement, right, before we actually start the project. So in the agreement, I'm going to quickly talk you through what was in the agreement. So each side needs to commit to a certain space, 2,000 square meters, a minimum. And then each side needs to raise their own funding, and they need to commit, dedicate personnel to the project. And every side needs to agree on the quality insurance. So for us, we later talk about the assembly standards, but we also make sure everyone send 100% base pair resolution correct DNA sequence to us. We agree on material transform. So we send a consortium, we don't exercise any IP on material. And then we agree on publications, how we swap authorships. But more significantly, we coordinate the co-submission. So recently, we have a big batch of papers we just co-submitted. And some sites, like later the paper was finished early, and she hold a paper for at least a year for us. So that was a big thing for the consortium, is we co-submit papers. And we support each other's grant proposal. We all agree there's no IP covering SC2 material, and that's really important. And it's freely shareable with the third party without MTA. And all the software developed in SC2 is open source under BSD. And we also give the ownership to individual site. So once you are given a chromosome, you own it, right? So we don't give the chromosome to the other site. So each site has the ownership. And then we have training and education. So we have been training people across site. And then we are complying with local laws. And there's a paper coming out last year on genetics talking about the freedom and responsibility. And this was mainly headed by Deborah Marshall, who is also here in the audience. We talk about the social benefits of SC2 and intellectual properties and safeties and governance. So I don't have much time to talk into it, but I just want to tell people a little bit about how we raise money. So in the beginning, the project was funded by NSF Savvy to start build a genome in Johns Hopkins. But after the first meeting in Beijing, Bija and Tianjin got $5 million from the Ministry of Science and Technology in China, and Imperial, headed by Tom Alice, got money from BBSRC to synthesize chromosome 11. And then when I moved to Edinburgh, we also got some money from BBSRC to do tRNA chromosome. But later on, Susan, Russell, and we got a bigger grant to support large piece DNA synthesis automation in Edinburgh called Edinburgh Genome Foundry. And then later on, we took on board Pasteur at EMBL, who has more specialized technology to the project. So Pasteur is headed by Lomeko Struer, who does 3C and 3D to look at the chromatin structure of the synthetic chromosomes. EMBL is using the tRNA-seq technology to look into the scrambled genomes. And then we got approached by two sites, which are Macquarie from Australia and Mashu Chang from Singapore National University. So they took on board 3 chromosomes and bring in their own funding from their local governments. So we have a lot of collaborations. So we train people across site. And then we share the protocols and expertise. So I just want to quickly show you 2 projects. So within SC2, we also have sub-projects. So this is a EROT grant, which we are going to do direct evolution at the genome scale. And this is between UK, Germany, France, and USA. And the other project, I basically tell you a little bit, is we delete all the tRNA genes. But my group synthesized all the tRNA arrays. So we make 16 individual tRNA arrays. We send that to different sites. And this has been quite a difficult project because, as you know, tRNA are really rich in sequence structure. I'm really pleased to announce it. We finished it this morning because as Singapore get the last piece this morning, we're done. So we got one single near chromosome in vivo, functional in yeast. And this really gives us an opportunity to work with all the 16 chromosomes. So we send 16 tRNA chromosomes. We test the complementation in different cell lines. And then just want to show you the interactons. So the green line means we have joint publications, and the purple line means we have joint grants. So if I'm also map the ongoing collaboration is going to be all over the place. But I just want to show you this is very active collaboration within the consortium. With that, I just want to point out there will be a meeting this year in Edinburgh, and you guys are more than welcome. Thank you very much.
Defining robustness is interesting to me in that the E. coli genomes that our groups have tried in the past are great in LB and minimal media, et cetera. When you get up to like 5% of a product that you're making in a bioreactor, the minimal genomes just don't stack up to MG1655. So how do you define robustness? And is this a yeast that can be used in commercial applications, or is it grow well in the lab? And how do you do that?
Right. So it grows well in the lab. Great. The scramble system, I think, is really your opportunity to explore a variety of minimal genomes under any condition you want. And those are ongoing experiments right now. And I think you can scramble, just to expand on what the scramble system is, we pepper the genome with LOX-P sites and then just express a cre recombinase. And that yields potentially massive recombination events and new genomes in each cell, and in parallel, a huge genetic variation. And so if one takes that population and selects under a condition of interest, one presumably could find anything that is possible biologically. And so I think that is a system that will yield incredible opportunity in commercial applications. Because one can scramble and then grow as one desires.
When you're comparing the different phenotypes, when you're comparing the different phenotypes in order to determine that you haven't affected phenotype, phenotype covers a very, very wide range of behaviors. How do you, with the yeast world even, try and assure that you're getting a large enough selection of comparisons?
Right. So we typically start with some very simple but robust assays, looking at just colony size under very stringent conditions, high temperature and non-preferred carbon sources. And then we go into more detailed analysis, including sort of RNA-seq and looking at the transcriptomics and proteomics in these cells, and evaluating genome-wide and proteome-wide the effects of the synthetic chromosomes. And we find subtle differences. And so it's a great question, what is fitness and what is wild-type fitness? We are working our hardest to test as many conditions as possible. One opportunity in yeast is to play it on a whole variety of different types of media that provoke different phenotypes given genetic backgrounds. And so we typically do those types of experiments as well. Matthew.
I have a question for Patrick.
Perfect.
Such a large international consortium, but what was the most challenging aspect of putting together an international consortium for such a scientific endeavor?
Actually, I got one more slide to show you. But really, I think the difficult part is really getting everyone to buy in. So you need to really tell each side what's the benefit, and having international collaboration coordination is absolutely key. I'm giving myself a pat in the back. But also the project needs to be well defined. So specific, measurable, attainable, and time bound. And I think that's why this consortium has been quite successful, is because the project needs to be well defined. And I think that's a challenge for HGP-write. We need to define the project, what it is, what's it for. And also, the other thing I think we did is really try to cultivate the culture. So if you ask people what's the culture at SC2, I want to say we are quite collaborative, inclusive, innovative, and responsible. I think it's try to cultivate the culture in the last few years has been quite difficult, and I think we have been quite successful too. Yeah. No more. Okay.
Sorry, just a quick question. I'm sorry if you said it. Is the mitochondrial genome also being edited?
Indeed it is, and a representative who's working on that is here, Sudarshan Pingle. And yep, it's undergoing designs. It's an interesting problem. It's about 90 kb and incredibly AT rich, which provides all sorts of opportunity for multiple designs and multiple, different builds to find out what works.
So this is my conflict of interest slide. And the question I'm asking is, why make millions of synthetic genomes, or billions, or trillions? And I mean this in all seriousness, and I think that's why we want to bring down the cost. So this is our progress curve for reading and writing. We are now at the point where we can make synthetic oligos for the entire human genome. In fact, I think this has already been done for various reasons: SNP analysis, expression analysis, and so forth. Assembling that's another matter, and that's this lower curve here. Let's see, here. There is a gap between the amount that we can synthesize. There's a very small gap between what we can sequence and what we can synthesize. So we can do multiple copies of human genome-sized things. But there's a gap in what we can assemble, and the gap becomes even bigger the more radical the changes we make in the genome, such as the ones that you'll hear much about today from yeast and E. coli. Making billions of genomes combinatorially is routine. It was fairly routine in 2009 when Harris Wang did this experiment. We put in the Nature paper that we had 4.3 billion combinatorial genome variants per day. And they weren't just random. These were really highly designed to optimize the ribosome binding sites in a pathway. If we go forward to today, we have 8 major tools for genome engineering and editing. They either use DNA, RNA, or protein as the matter that goes and finds the needle in the haystack, finds that one base pair out of 6 billion in the human genome and edits it. Even though we have crystal structures for most of these, we don't actually have one for lambda beta, to my dismay. We have a ways to go before all of them are as easy to manipulate. This project that Nili Ostrov will present in a moment started where we changed one genome, one codon genome-wide in E. coli, and we achieved in that project a very efficient use of non-standard amino acids, biocontainment, that is to say genetic and metabolic isolation, and to our surprise, multivirus resistance. 4 different classes of viruses that we analyze in detail are 100 to 1,000-fold down. And this was one codon, and then Nili will tell you about the 7 codons, which is a team effort. That is genome-wide. I mean, that happened to be a rarer codon. As you go to more and more codons, you really need to do it genome-wide in a genome which is composed mostly of proteins. But as we get to higher, larger eukaryotic genomes, like a mouse, pig, human, and so forth, the exons that compose the protein coding regions become sparser. They're about 1% of the genome, although it might be most efficient to engineer them with the 100 kb swapins that Jeff described. But these are examples of sort of coding region level changes where you might want to change the species or the viral resistance via recoding. But we've heard how, for the most part, for both yeast, and you'll hear soon about E. coli, that we've left the non-coding regions intact because we don't understand it well enough to engineer it. But that has to change, and I think that's one of the things that we hope to do. And to just show you that even things that seem to be junk DNA, like ALU repeats, have examples where a single point mutation in ALU repeat can cause a change in transcriptional activity and be associated with a leukemia. So we want to engineer the repetitive elements genome-wide, and Luhan Yang will tell us something about that later on. But not just the repetitive elements, but every base pair, because there are chromosome folding rules we know nothing about, and we can multiplex, we can put together a large number of changes into a single genome, where if you make a heterozygous for every cis-regulatory mutation, and you can measure allele-specific expression, you can essentially do 20,000, 30,000. There are possibly hundreds of thousands of different promoters scattered throughout the large plant and animal genomes, and we'd like to be able to do that. We can do them all at once, at least. And so the Encyclopedia of DNA Elements is based on observations of all these epigenetic changes, RNA and chromatin changes, and there is transcription almost throughout the genome. There are binding proteins binding all over the place, and one of the critiques of this project, it's a wonderful project, one of the critiques is just because something binds, just because something is expressed at the RNA and or protein level doesn't mean it's functional. And so to do that, we need the organoids that Alex will talk about as well, in order to assess the changes to every base pair and try to do as many base pairs at once. We can make ancestral genomes, not just by reading ancient DNA, but by actually constructing phylogenetic trees. So I'm going to get us back on schedule and give time for discussion. I don't even think my 2 minutes.
Do you want to take a couple of questions?
Yeah, sure. Yeah. If there are any. Yeah.
George, can you elaborate on these promoters? We went too fast.
So the idea is that there's some investment you put into analyzing. If you wanted to treat the entire genome as a cis-regulatory element, right? And even the coding regions we know in some organisms can be cis-regulatory element. So you've got 3 billion, let's say, for mammalian genome points that you want to change. And you'd like to, and there's an overhead of analyzing that. So let's say you want to change one base pair and want to ask, does it affect the liver, the brain? There's many cell types. You might want to do a serial section through a mouse embryo and see all the tissues that are affected. Well, if you have allele-specific RNA expression, you can, for example, or any allele-specific epigenetics, you can see, and you made an allele-specific mutation, then you can see how it affected one allele relative to the other. You have a beautiful internal control, which is the other chromosome in a diploid. Since, to some extent, those cis-regulatory elements, if they're subtle changes, they're independent of one another. So this mutation on gene A is not really influenced, in a heterozygous state, is not influenced by heterozygote mutation in gene B. So in principle, you could change all the genes subtly in heterozygote state and read them all out in parallel in every cell in the body of the animal or plant.
But many of these cases won't be independent from
If you greatly overexpress or knock out something that's haploinsufficient, you will have impact on other. But if you do enough of these things, I mean, you're committing to 3 billion of these things, so in many combinations. Yeah.
Comment on how much of epigenetic editing we'd want to do, so you could actually attach TCAS9 to, say, TET1 enzymes or DNMTs, and think about actually both genetic and epigenetic targeted modifications.
I think that's a very important part of this project for 2 reasons. One is you need to be in control of the epigenetics either for certain organisms where it's hard to create the whole organism, like say a giant sequoia or a human being, but also so that you can test that you understand the impact of mutations and epigenetic changes. Alex will talk more about this, about how you can make various different organoids and how you can systematically go through epigenetic factors. For example, we now have a complete collection of all the human transcription factors, and you can go through those now systematically in various combinations. I think this is one of the coolest part of this project, is being able to come up with high throughput ways of testing developmental biology. Ideally, some of it can be done in a few microns rather than having the expense of a mouse facility or a bowhead whale facility. Okay, thank you very much.